So today I'm going to tell you a little bit what we've been busy doing during the pandemic on our favorite molecule, which is Tetraspanin CD82. I have no conflicts of interest to declare about this work. And so tetraspanins are an interesting family of molecules. They are proteins that organize protein complexes at the membrane. So in some ways, we can think of them as chaperones that are busy taking things in and out of the membrane all the time. Their binding partners can change in different tissues and with cellular states. So obviously, this is a very dynamic process. And so it can be that during proliferation, for example, or differentiation of cells, different binding partners are linked to tetraspanins. One of the signaling molecules or family of signaling molecules that are known to act through tetraspanins are the integrins. And originally, CD82, which is the tetraspanin that we focused on, was mostly researched in cancer. And in cancer, having high levels of expression of CD82 is usually associated with better prognosis because it was shown that it's a non-metastasis suppressor. So when CD82 is high, it's less likely that the tumors are very aggressive because it's an anti-adhesion molecule. And so our work on CD82 started several years ago, and this is all published, and I'm just going to go over very quickly. We showed that in human satellite cells, CD82 is an excellent prospective marker for isolating myogenic progenitors. We also have shown that it binds to alpha-7 integrin and alpha-sarcoglycan. When CD82 was silenced in human cells, we saw that there was a defect in cell proliferation. And then in cell lines from patients with muscular dystrophy, CD82 was significantly decreased. And so all of this work prompted us to study the significance of this molecule a little bit more in normal and in dystrophic background. And so what we did not know at the time was whether the loss of CD82 expression in satellite cells affects their function. And whether loss of CD82 can change the pathology of dystrophic muscle as well as normal muscle. Additionally, based on what I told you already on tetraspanin function, we wanted to know whether the binding partners of CD82 change in proliferation versus differentiation of muscle stem cells, and whether the expression of CD82 is affected in dystrophies other than DMD, which is what we originally studied. And so I'm going to go over all of these questions in 10 minutes or 15 minutes, whatever I have left or less. And I'm going to tell you a little bit more, some recently published data, as well as some unpublished data in my lab, for which I hope that you guys are going to give me some feedback, because that would be fun. So what we did originally was to actually import a mouse that was actually full knockout mouse from a different lab, and we actually analyzed the muscle of these mice, both at 4 months of age in younger and older mice, and we saw that these mice basically they were not really affected, as you can see from the histology on the left side of the slide, as well as all of the analysis that we performed, both histologically as well as functionally. These mice did not show a lot of differences from wild-type mice that were age-matched. When we actually injured the muscles of these mice using cardiotoxin, we actually saw that something was starting to happen, where the CD82 knockout mice actually had a dramatic and significant shift to the left, and the myofibers were significantly smaller in the CD82 knockout, both at 7 days and 21 days following regeneration. And so based on that, we presume that some defect either in satellite cell activation or in satellite cell fusion could be occurring, because these are the two basic biological phenomena that are important during muscle regeneration and formation of new myofibers. And so our first steps were to actually count the number of PAX7 positive cells in CD82 knockout mice, both at a steady state, as well as in regenerative conditions. And in both cases, we saw that the number of satellite cells was not significant significantly different in uninjured as well as in injured mice. And so based on this data, we actually started presuming that perhaps the activation of satellite cells was impaired. And so we did this in different ways. The first way was to actually isolate the cells in non-injured mice. And we actually counted the number of PAX7 positive and MyoD positive cells, which would be the activated cells, versus PAX7 positive MyoD negative cells, which are the quiescent satellite cells, at 24, 48, and 72 hours following isolation. And so what we saw, if you follow this panel C, was that the CD82 knockout mice, which are depicted right here in green, actually have a lot of PAX7 positive MyoD negative satellite cells, while the activated satellite cells were actually decreased, significantly decreased compared to wild type. You see in the wild type here, at 24 hours, we have the same non-significant difference number of quiescent satellite cells, but then they decrease rapidly. And at 72 hours, nearly all of the satellite cells from these mice are activated and double positive for PAX7 and MyoD, while the CD82 still retain a large proportion of them, the PAX7 positive signature, and they were MyoD negative. While for the PAX7 positive, MyoD positive is the exact opposite. We also did this in a little bit of a longer time point, and we actually looked at a different marker. We looked at the co-expression of PAX7 and Ki67, and we were actually able to reproduce the same type of findings, where the CD82 null mice, the satellite cells extracted from these mice, actually retain PAX7 expression, while the activation was actually decreased. And so we thought that because of these findings, it would be interesting to see what the effect was of ablating CD82 in a dystrophic mouse. And again, this data was recently published. And so we compared MDX5CV mice, which is a low revertant model of MDX, with double null mice, both at 4 months as well as 1 year of age. And you can see in this image, how the mice that have ablated CD82 in the dystrophic background, they present with severe kyphosis, which was also accompanied by a significantly increased inspiration rate with plethysmography. And you can see how the fibrosis is significantly increased in these mice, as well as presence of fat in the muscle and intermyofiber fibrosis. And in these mice, we also compare the size of the myofibers as well as the strength. And so you can see on the left panel how by eccentric contraction, the CD82 MDX mice were significantly weaker than MDX mice pretty much after the second contraction occurred. And so these mice are definitely weaker, and this is in very young mice that we were seeing this. And then when we compare the myofiber size of the mice, the double null mice, which are in red, versus the MDX5CV, which are in blue, versus the CD82 knockout that are actually in green, you can see how significantly smaller the double null mice have the smaller myofiber the double null mice present compared to MDX and compared to CD82 null. And we did this in many different ways. And so we also looked in the dystrophic background, because it was so much more severe, whether the satellite cells could activate or not, because we know that the dystrophic background it undergoes degeneration and regeneration continuously. And so we thought that this might change a little bit the dynamics of satellite cell activation. And what we saw was that it was still consistent with what we saw in the CD82 null mice, where clearly even in the dystrophic background, there was a reluctance of satellite cells to activate. However, it was not as dramatic as we saw in CD82 knockout compared to control, because it was declining, and at 72 hours, they were basically going back to baseline level compared to the normal dystrophic background. However, we did see a little bit of a reluctance to activation, and we don't know if this is the only phenomena that actually can contribute to a worsened dystrophic phenotype in these mice. And so all of this data is published. And so, you know, we wanted to go a little bit further and try and understand a little bit more what CD82 is actually doing in satellite cell, as well as possibly in myofibers, just because of the differences in possibility in binding partners that CD82 might have. And so with the rest of the time that I have, I'm going to give you a little bit of history and results of new work that is not published, and hopefully we can have a little bit of discussion on the new data together. So the first type of work that we did was to actually try and identify the binding partners of CD82 in myogenic cells. And we did this by immunoprecipitation following by mass spec. And so what we did, we took human cells, these are primary human cells, under proliferation condition on the left side and under differentiation condition on the right side. And we identified by mass spec the binding partners of CD82 that were immunoprecipitated with the CD82 antibody. And so at first glance, what you can see is that they are different. Some of them are there, both in proliferating and differentiated cells, but many of them are different. And one of them that was there in both is the second one on the top in the proliferating cells is called LMN1. And it's also there in the differentiating cells. But then in the differentiating cells, we also saw some other interesting binding partners that we pursued a little bit more. And one of them is dysferlin and myoferlin. And you can also see annexing A2, which we all know are important for dystrophic muscle function. And so what we did was to start studying by immunoprecipitation, validating that there was indeed binding of these markers to CD82. And in the first Western blot and confirmatory immunoprecipitation on the left, you see how we did these studies on LMN1, and actually this confirmation failed. And so when we actually did immunoprecipitation using CD82, an antibody to CD82, and then we blotted for LMN, we saw that actually there was background staining also in the IgG control. And so we discarded this as a true interactor, and we did not follow up on this anymore. And we also then continue to validate our findings on other potential binding partners, and we validated this with dysferlin and myoferlin. And these were actually much more convincing, where we saw that immunoprecipitation of CD82 actually confirmed that dysferlin was pulled down using a Western blot for dysferlin, as well as myoferlin. We actually did also reverse IP in this case, and they were also confirmed. And the second method of validation that we used was to use co-localization of these molecules by immunofluorescence. And you can see this on the bottom panel. You can see in red immunostaining for CD82 in both individual mononuclear cells. And some of these co-stain with dysferlin that you can see in yellow in the merged images on the right. Both in mononuclear cell as well as in myotubes that you can see on the bottom. And so CD82 does bind, according to our biochemical data, to both dysferlin and myoferlin. But the big question is, what is the relevance of this binding in dystrophic muscle, particularly in dysferlinopathy? And so the
5 minutes left.
Yes, the first thing that we did was to actually test whether expression of of dysferlin was actually changed in CD82 knockout mice. And we actually saw by a Western blot that this was indeed true. It was significantly decreased in CD82 knockout, while myoferlin, if you look at the middle panel, did not change at all. And then we actually did the opposite experiment, where we took blood J mice. which are a primary dysferlinopathy model. And we actually tested whether CD82 expression was decreased, and the answer was no. So basically what we saw was that in CD82 knockout mice, dysferlin was changing, but conversely in dysferlinopathy mice, CD82 was not changing. And then we moved this study into humans, and this is where things became very interesting. You can see on the left side in A and in B. In A, we have these are primary cells from control, which were stained by FACS for CD82 and CD86. CD82 is on the x-axis and CD56 is on the y-axis. So the double positive cells that you see right here are clearly myogenic cells in our plots. And you can see here a DMD sample where the myogenic cells are decreased in percentages, but they're definitely there. And down on the bottom, J309 and J322 are actually two patients with dysferlinopathy. And there is absolutely zero expression of CD82, although CD56 is still expressed, which is the other marker for myogenic cells in human. Then, courtesy of Vincent Mouly, he actually gave us additional samples for dysferlinopathy and additional control using cell lines. And so you see AB1190 and AB1079. Those are two control cell lines, and you can see there is 100% co-expression of CD56 and CD82. These are myogenic cell lines that he generated and kindly makes available to all of us in the field. And so we have co-localization of these two markers. And then ER and RB are two additional dysferlinopathy patients. And you can see that in ER, CD56 is not expressed while CD82 is expressed. And in RB looks completely normal. So the J309 and J322 are two patients from Brazil. And we try to confirm actually the reduced expression of CD82 by Western blot that you can look at in C. And you can see that there is exactly correlation between the finding that we had by FACS with the Western blot, where these two particular patients actually show severe decrease of expression of CD82. Again, we confirmed this data by immunofluorescence that you can see on the bottom, where all of the controls were actually showing strong and robust expression of CD82 intracellularly in vesicles, while the dysferlinopathy patients, the Brazilian patients, did not. And instead, ER that you can see on top actually show co-expression. So where does that leave us? And that's where I would like to start some of the discussion. We see that we've seen that CD82 favors satellite cell activation. I have shown you that loss of CD82 in dystrophic muscle worsens the phenotype. And I hope you are convinced that CD82 is one of those proteins that can bind different protein partners in myoblasts and myotubes. And we have a lot more to learn about this. And also, what I have shown you, and I hope you are convinced by that, is that expression of CD82 at the cell surface can be affected in dystrophic cells. And there are some dystrophies that can be affected in different ways. So could this be a biomarker for dystrophy? Could this be a biomarker for satellite cell function? We don't know yet, but I hope to stimulate some discussion about this, and certainly we have lots of work ahead of us to continue understanding what is the function of these molecules in both myofibers as well as satellite cells. So briefly, just to conclude, I want to acknowledge all the people in my lab who have done this work, particularly Tatiana, Ariel, and Alec in my lab. And then, of course, all of the collaborators that have helped us along this journey, and particularly Maritz Beinzoff and Mayana Zas, who contributed the Brazilian patients to our lab, and Vincent Mouly, who contributed the cell lines. And then, of course, the funding agency without whom none of this work is possible. And so I'm happy to start the discussion and take any questions that you may have. Maybe you can put us on track on a good road to understanding more about this.
No, I would just have one. You're going to change crosstalk because CD82 is also, and dysferlin are both very much part of the extracellular vesicles.
Absolutely, and we have seen co-localization of CD82 in extracellular vesicles, but what we have seen is that CD82 expression does coincide with dysferlin and myoferlin in the vesicle, but there are also some unique vesicles that are only labeled with CD82, and so again, it's a partial overlap, and some of this needs to be teased.
Yeah, but if your CD82 is downregulated, this could have an effect on pathology.
Yes.
You'll change your trafficking. Yeah.
Yes.